Hello everyone, my name is Shanni Li. Today I'm going to present my project, which is regarding the in silico discovery of specific chip drug-like ligands. This project was conducted under the supervision of Dr. Shapira using ICM Pro software by Microsoft. Chip protein is an E3 ubiquitin ligase that is encoded from stop1 gene. It can present as a monomer or a homodimer structure. Chip protein was found widely expressed in various human tissues, including heart, brain, kidney, lung, and fat. A monomer structure of chip consists of three domains, which are TPL1, TPL2, and U-box. TPL1 is a N-terminal tetracyclopeptide repeat 1, and that is labeled in light blue. TPL2 is a coin region that is labeled in orange. The U-box domain is a C-terminal domain of the protein that's labeled in purple. CHIP has several biological functions. Its main function is to maintain protein quality control using ubiquitin proteasomal system. In addition to this, it can also regulate some other cellular processes, including cell proliferation and growth, cell apoptosis, and drug metabolism by CYP3A4. CHIP has a wide range of substrates, such as heat shock protein 70 and 90, SMAT1 and 5, and MEKK2. Most substrates, but not all, bind to the TPL1 domain of CHIP, which is the region colored in blue. As shown in this figure, the ribbon represents the substrate that interact with the TPL1 domain of CHIP. Since CHIP plays an important role in regulating protein quality control, and many previous studies have determined its ability to mediate misfolded or aberrant protein for degradation. It has been identified as a promising pharmacological target for drug development to treat diseases such as Alzheimer's disease, Parkinson's disease, and stroke. Its role in managing neurological disorder is more pronounced, as it has been shown the ability to predict beta-amyloid, alpha-synuclein, as well as phosphorylated ELF2A and AKT4 degradation. The goal of this research project are to discover putative drug-like ligands that can specifically bind to the TPL1 domain of CHIP, as well as to select drug-like CHIP ligands that can be used to make PROTACs for target protein degradation. A PROTAC is a bifunctional molecule that refers to proteolysis targeting chimeras. It is evolving as a novel therapeutic strategy that proposes to treat diseases such as cancer and neurological disorders. A PROTAC consists of three components, which are an E3 ligase ligand, a protein of interest ligand, and a linker. As the name implies, the E3 ligase ligand and the protein of interest ligand bind selectively to its E3 ligase and protein of interest respectively. Its mechanism of action is illustrated in the figure below. Upon the binding of a PROTAC to its selected E3 ligase and protein of interest, a stable ternary complex is formed, and that enables the E3 ligase to ubiquitinate the protein of interest for degradation due to their close spatial proximity. The current developed PROTACs, which can recruit VHL, CRBN, IAPS, and MDM2 proteins, are proposed to target oncoprotein for degradation by 26S proteasome. Since there is no available PROTACs to target CHIP, It is therefore important to discover drug-like chip ligands that can be used to develop PROTACs. The study rationale is based on the ability of chip to bind to the HSP90 protein. Chip can bind to its substrate and initiate either direct or indirect polyubiquitination. Figure A is showing the indirect polyubiquitination. This requires the binding of HSP70 or HSP90 to the TPR1 domain of chip. This enables chip to ubiquitinate the misfolded protein that is attached on the HSP70 or HSP90 for degradation. Additionally, direct polyubiquitination is shown in Figure B. In this case, the SMAD1 protein bound to the TPR1 domain of chip is directly ubiquitinated by chip for degradation. Overall, the polyubiquitination path is dependent on the substrate that binds to TPR1 domain of chip. The project was started with the analysis of interaction between chip and the endogenous peptide HSP90, and then followed with potential hotspot identification by fragment docking screening. And next, two templates was used to conduct virtual screening to discover hits that are predicted to interact with chip. One template was added from the endogenous peptide HSP90, and the other was added from a synthetic peptide that was identified by another study as a high binding affinity ligand for the TPR1 domain of chip. And also, two types of virtual screening were performed in this study. One is a ligand-based right search, which serves to discover hits that have similar atomic property field to the template. And another is receptor-based docking screening, which is used to discover hits that can fit well into the receptor pocket. The output hit list obtained from the right search was then used as the input data to conduct the docking screening. And lastly, the hit was selected based on some parametric score generated in RAI and docking screening. Here are some important results obtained in this study, and they will be elaborated in detail in the following slides. In the analysis of protein-ligand interaction, HSP was found binding selectively to the chip TPR1. Although there are 3 binding pockets identified in the chip TPR1, HSP90 interact with only 2 of the 3 binding pockets. The side chain of M505 and V508 in the HSP90 form hydrophobic interaction with the 2 binding pockets, as the intermolecular distance between the side chain of HSP90 and chip were measured to be less than 4.0 angstrom. Aside from the hydrophobic interactions, 2 weak hydrogen bond interaction was found. One is between E506 and K73, and another is between D509 and N66. The 2 hydrogen bond interactions are weak because the intermolecular distance measured between the 2 heavy atoms, which are the nitrogen and oxygen, are greater than 3.0 angstrom. These results of the study are consistent with that of the previous studies. which have reported that the MEEVD motif of HSP90 plays an important role in the binding of HSP90 to the CYP-TPR1. The MEEVD motif is illustrated with the X stick, showing this part. The residue in the CYP-TPR1, which are frequently involved in the interaction with the fragment molecules, are phenylalanine, isoleucine, leucine, valine, and lysine. Fragment molecules were capable of binding to this binding pocket in the CYP-TPR1, whereas the HSP90 occupies only 2 of the 3 binding pockets of the CYP-TPR1. In one binding pocket, the side chain of the residue M505 in the HSP90 interact with the residue F99, F132, I136, and V95 in the CYP-TPR1. Furthermore, the side chain of V508 in the HSP90 interact with the CYP-TPR1 residues F38 and L69 in another binding pocket. These 2 binding pockets are occupied by both the HSP90 and the fragment molecules docked to the CYP protein. The analysis of fragment docking results show that the residue F99, F100, F132, I136, L69, and V95 are frequently involved in hydrophobic interaction with the fragment molecules, whereas the K96 in the CYP-TPR1 are frequently involved in forming hydrogen bond interaction with the fragment molecules. Among the listed residues, F99, F100, F132, I136, L69, and V95 play important roles in forming hydrophobic interactions with the fragment molecules, and the hydrophobic interactions is the main joining force that facilitate drug receptor interaction. HSP was edited into an optimized template named as template 1, and a synthetic peptide was edited into another optimized template named as template 2. Both templates were edited based on the hydrophobic interaction of their original peptide with the CYP-TPR1. Because hydrophobic interaction is the main driving force for drug receptor interaction, template addition was mainly based on the hydrophobic interaction. After the addition, both hydrophobic ends of template 1 and template 2 occupy the binding pocket of CYP-TPR1, and it is the place where they form hydrophobic interactions with the residue in the CYP-TPR1. For both template 1 and 2, the result of ligand-based RI and receptor-based docking screening provide a list of molecules that have no found correlation between their RI and docking score. Generally, the promising hits have both high positive RI score and low negative docking score. But in this study result, most hits have docking score ranged from -10 to +10, and they all have a RI score approximately to 0.75. Therefore, his selection was performed with write and docking results separately. 3 hits with the highest write score were chosen as representative example for protein-ligand interaction analysis. The receptor protein used in the virtual screening is the mouse chip. The 3 hits were found commonly to form hydrophobic contacts with the residue F99, F132, L69, and F38 in the chip TPR1. The aromatic rings at both ends of the molecules participate in either parallel or perpendicular pi stacking with the aromatic ring of phenylalanine. Except for the hydrophobic interaction, there was no hydrogen bond interaction observed between the hit and the chip TPR1. Three hits with the low negative docking score were chosen as representative example for protein-ligand interaction analysis. Similarly, these 3 hits form either parallel or perpendicular pi stacking with phenylalanine in the chip TPR1. In addition to the hydrophobic interactions, they also engage in forming H-bond interaction with lysine 96, as the intermolecular distance measured between heavy atoms are less than 3.0 angstrom. After virtual screening was completed with template 2, 3 hits with the highest write score was selected as representative example for protein-ligand interaction analysis. Here, the crystal structure used in the virtual screening is the human chip instead of the mouse chip. Although the amino acid sequence of human chip is 98% identical to that of the mouse chip, the position of phenylalanine is labeled in a number different from that in the mouse chip. Except for the different numbered position, these 3 hits form hydrophobic contacts with the chip TPR1 in a manner similar to that observed in the previous representative hits. One intermolecular H-bond interaction was found in hit 2, but there was no any intermolecular H-bond interactions was observed. Here, 3 hits with the low negative docking score were selected. The first hit formed a strong H-bond interaction with chip TPR1 in addition to hydrophobic interaction with phenylalanine. The hit 2 and 3 have no formed H-bond interaction, but rather engage in parallel or perpendicular pi stacking with the aromatic ring of phenylalanine. For the purpose of hit selection, the correlation between the docking and RT-CNN score was determined using a scatter plot. 2 hit lists were generated from the virtual screening. One is based on template 1 and the other was based on template 2. For both hit lists, there was no remarkable correlation found between the docking and RT-CNN score. The first 3 hits with the lowest docking and RT-CNN score were identified in the scatter plot. Each dot is represented a hit. For the plot in the left, the 3 hits are identical to the top 3 hits listed in the docking result from template 1. For the plot in the right, the 3 hits are identical to the top 3 hits that are listed in the docking result from template 2. Here are the summary table containing the representative heat mentioned in the previous slide. Table in the first row indicated heat with high rise score. Table in the second row contain heats that have the low negative docking score. The right docking and RTCN score are important parameters to be considered for heat selection. Although two structurally different templates were used for virtual screening, of the top heat selected from the result of virtual screening have relatively the same rise score at a range of 0.71 to 0.85. This indicated that the top heat have a highly similar atomic property field to the template used for the virtual screening. Unlike the rise score, the top heats do not share the same docking and RTCN score, and this difference can be used to predict heat ligands that can better interact with the receptor chip. The top heat selected from research are observed to have similar Ri and RTCN score, but a highly varied docking score. This implies that they may form induced fit clash with the residue surrounding the receptor binding pocket. Since the docking score is used to be penalized by the presence of clash. In addition, it was also noticed that some heat are more likely to have a positive docking score if they consist of multiple or complex rigid aromatic ring structure that are used to interact with the binding pocket of chip TPR1. Since the larger rigid component could have a higher propensity to occupy other areas aside from the binding pocket, the positive docking score of the heats may be accounted for by their out-of-pocket binding due to the complex aromatic ring structure. As the ligand's ability to fill the binding pocket space is a fundamental requirement for drug design, it is necessary to consider the out-of-pocket binding of the heat to the receptor for heat selection. So in conclusion, although the top heat obtained from the virtual screening were based on the structurally different template 1 and 2, they all have the ability to form hydrophobic interaction with the side chain of phenylalanine and one binding pocket, as well as form hydrophobic interaction with the side chain of phenylalanine and leucine and another binding pocket of chip TPR1. Also, it's worth noting that a single or multiple aromatic rings are commonly found at both ends of the heat structure, which participate in forming a parallel or perpendicular pi stacking with the side chain of phenylalanine. This commonality implies that the aromatic interaction can be used to predict the interaction of the putative drug-like heat with chip. Because aromatic ring-related hydrophobic interactions are widely found in chemical and biological systems, and also around 50% of pi stacking interactions are identified between the aromatic rings in ligands and in phenylalanine of the receptor protein. The hydrophobic interaction should be considered for heat selection. In comparison to the rise score, the heat that are selected based on the highly negative docking score appear to frequently form a strong hydrogen bond with the lysine at position 96 in the mouse chip or at position 95 in the human chip. This observed frequently hydrogen bond interaction with lysine is consistent with the fragment docking result, which was indicated that over 60% of a total of 1,600 fragment molecules were observed to interact with lysine 96. This suggested that lysine 96 might be a hotspot residue that facilitates the binding of the hit to the receptor chip. In addition, hit with rigid aromatic rings are more likely to have positive or less negative docking score. Also, hit with high right, low docking, and low RTCN score have relatively less rigid or complex aromatic ring structure. So there are two limitations in this study. One limitation of the study is that there are only two templates used for the virtual screening. One is edited from the endogenous peptide HSP90, and the other is edited from a synthetic peptide. As TRPTPL1 can also interact with other proteins such as HSP70 and SMAT1. Future studies should also make templates from this protein that are capable of binding to the TRPTPL1 to perform virtual screening for his selection. Another limitation is that no statistical test was used to determine the frequency of hydrogen bond interaction between the hit and lysine 96 in the mouse chip or lysine 95 in the human chip. Therefore, further study is suggested to analyze if the lysine 96 or lysine 95 is significantly involved in the hydrogen bond interaction with the hit. For the hit list generated based on template 1,000 hit with highest right score and 1,000 hit with the lowest docking score should be selected to test in vitro experiment for the next step. Also, the same criteria that are used to select hit from the template 1 should also be applied to select hit from the hit list generated based on template 2. Although many of the selected hits may have a positive docking score due to the penalty for clash or out-of-pocket binding, it is still worth investigating their interaction with the receptor chip, because the target binding affinity and efficacy need to be confirmed with the in vitro study. This is the end of my presentation. Thank you for listening.